Now we still have that blastocyst that we were just looking at. Here's that inner cell mass that's going to become the embryo. And here's the trophoblast. And we see that the trophoblast has progressed and is further along in implantation into the endometrium. So the villi have gotten bigger. They've invaded further into the endometrium. Now, as they invade into the endometrium, they are breaking down the endometrial tissue ahead of it. And among the tissues that are being broken down are the small blood vessels in the endometrium. And when we break down the walls of those small blood vessels, they bleed. The blood escapes, and we'll start to see accumulations of blood in the endometrium. And these accumulations of blood are in these little spaces that are called lacunae, or lacuna spaces. Okay, so the blood that we see in the lacunae is maternal blood that has escaped from the mother's uterine arterial system. So here's a branch of the uterine artery that's come in and supplying the endometrium. As the blood from those vessels escapes, we see it accumulated in the lacunae. Now notice that the lacunae are found in between the villi. That's where these accumulations of maternal blood are, in between the villi. After a short time, we will start to develop inside the villi, that will start to develop embryonic blood vessels containing embryonic blood cells. So inside the villi, we'll start to develop embryonic blood vessels with embryonic blood. Outside the villi, in the lacunae, we have maternal blood. So we have embryonic blood here, we have maternal blood there. The two blood systems are separate, so you don't get a mixing of the blood cells. But there's only a few cell layers separating them one from the other, so because the separation is so thin, that provides the opportunity for the movement of small molecules across from one blood system to the other. So you can get oxygen from the maternal blood into the embryonic blood. You can get CO2 from the embryonic blood into the maternal blood. You can get glucose into the embryonic blood, and so forth. Okay? These embryonic blood vessels that we see here in the villi will eventually connect with blood vessels that form inside the embryo. And those blood vessels and these blood vessels will be connected by blood vessels that pass through what becomes the umbilical cord, and that's what we'll end up calling the umbilical artery and umbilical vein. So that's connecting the blood vessels out here with the blood vessels in here. And this out here is what it obviously becomes the placenta. So the fact that there are some things that can get across from maternal blood into embryonic blood and other larger molecules that can't get across, that serves as the basis for the placental barrier. Some things can get across and some things cannot. But we now have the mechanism for supplying the embryo with oxygen and glucose and so forth. Okay? So that's all that we will say about the placenta. Let's now turn our attention to the embryo developing here in the embryoblast. During the second week, what we see happen is that the cells of the embryoblast differentiate and arrange themselves into two layers of cells, two layers side by side here. And that's why we call this the two-layered embryo. So if we take a look at this in the larger view, here it is here, the two layers of the two-layered embryo. Now, what I want to do on our next slide, we're going to take a look at this very same image, but we're going to rotate the image 90 degrees. So instead of the two layers being side by side, right and left, I'm going to rotate it 90 degrees so one is above the other. And that's what we'll see on this slide. It's the same image, but rotated. And I did that for two reasons. Number one, by convention, whenever you're looking at a section through an embryo, it'll always be oriented this way, with the two layers above and below. And the other reason is that the names of these two layers will be based upon this orientation, the names that we assign to them. So the layer that is now the upper layer is the layer that we call the epiblast, epi meaning upon or above, and the layer that's below is called the hypoblast. Hypo means below. So this is the hypoblast, that's the epiblast, the two layers of the two-layered embryo. Now, those two layers are separate. They're separate from one another, two separate layers. sort of like two pancakes, if you think about it, two pancakes, one lying on top of the other. You can separate them and create a space in between them. And that's true everywhere except one place, where the epiblast and the hypoblast are fused together, just in one location. Every place else they're separate. Here it's separate, here it's separate, but right there they're fused together. Okay, it's as if you took your fingers and squeezed them together at that one point. At that point where they are fused, we call that the procaudal or the precaudal plate. You'll see it both ways, precaudal or procaudal plate. That's where the epiblast and hypoblast are fused together, where there is no cavity in between them. The procaudal plate is going to be the place where the future mouth will develop in the embryo. That's where the mouth is going to develop. Well, that turns out to be a very useful landmark for us, because if we know that this is where the mouth is going to develop, that tells us that this part of the embryo is where the head is going to develop, because the mouth is in the head. So that means that once we know that this end of the embryo will become the head, which tells us that this end will become the tail. So we now have established some polarity to the embryo. Okay? Before recognizing that, we couldn't tell which was the head end and which was the tail end, but once we see the procaudal plate, we know that this is going to be the head end, and therefore this will become the tail end of the embryo. So we now have established polarity. Okay? So that's what occurs in the second week of development in the embryo. The inner cell mass differentiates into a two-layered embryo, an epiblast and a hypoblast. And those two layers are fused together in one place, and that's called the procaudal plate. Now, to sort of give you a sneak preview of what's to come, I can tell you that all of the future cells of the embryo, all of the cells that will give rise to our adult cells, will come from the epiblast. The hypoblast will give rise to nothing that survives to the adult. So there are no derivatives of the hypoblast. Hypoblast cells are all going to die. Everything that will persist will have arisen from the epiblast, and we'll see how that happens in just a moment. Okay? So that's the story of the second week of development. In the third week of development, what's going to happen is we're going to convert this two-layered embryo to a three-layered embryo. So that's a useful mnemonic. Remember, you have a two-layered embryo in week 2, which will become a three-layered embryo in week 3.